The first topic will be with respect to biotechnology. Second, we'll move towards health and disease. Next part is your space technology. Next comes the information and communication technology. Next comes a topic with nuclear technology. And finally, last but not the least, is the defense technology. Hello everyone and welcome to PW Only IS. Friends, today we are here in order to discuss few of the important topics under science and technology. As we all know that if we see a particular trend of various subjects that is being asked in the prelims of our UPSC, definitely you will see a range of like 20 questions, 15 to 20 questions in few of the subjects like Indian polity, economy and geography in the recent days, not in the earlier days. With the past 2 years, you can see 2023 and 24, there is a spike in the number of questions with respect to these subjects. But whenever you are asking for the science and technology, definitely it comes around 10 to 12 in each year. You can see that constantly 12 questions has been asked in 19, 20, 21. 22, in 22 there are 14 questions, and in 23 and 24 there are approximately 10 questions that we can count under the subject of science and technology. So in this very session, I will try my best to incorporate the maximum current affair portion that is being around us in the past year or in last one and a half years. So starting with the very basic topic, or let me summarize the basic topics that we are discussing now. The first topic will be with respect to biotechnology. And with the biotechnology portion, second we'll move towards health and disease. Altogether, if someone asks that what is the weightage of these two topics, you can say that approximately 3 to 4 questions per year will be asked from these two chapters itself. Why I am combining these two chapters? Because they are interrelated. Next part is your space technology. As you all know that ISRO is doing its great. Means everywhere you will see the news flashes with ISRO achievements. There are new targets that is being achieved throughout the year. What were they, or what about them? All the space missions. So space technology comes in important section. Next comes the information and communication technology. As you all know, with the advancement of artificial intelligence and with the advancement of quantum computing, this ICT, information and communication technology, is again in the news throughout the year. Definitely we'll catch up with all the news. Next comes a topic with nuclear technology. Nuclear technology is something which is exceptionally important this year. As we all know that in this particular segment in nuclear technology, from the past 30 to 40 years, we were trying our best to reach second stage nuclear power program. But last year in 2023, last or in the beginning of 2024, we see the ray of hope turning into reality. And for the very first time, which should have been done in 1980s or 90s, we have achieved in 2024. That is why this phenomenal advancement in the nuclear technology has made this topic very, very important for this year prelims. Nuclear technology, we will see into it and we'll we'll definitely cover these topics as well. And finally, last but not the least, is the defense technology. Defense technology, as you all know, with the recent days news, the defense technology is already in news. But I think that the UPSC is going to ask the questions till April at max, and till April we could not find any such defense technology that is being currently in news. Just for example, I would say BrahMos. BrahMos was earlier in news as well, because BrahMos new generation is being made for the export version. And just like now, there are S-400 which were in news earlier as well. So these questions might come, but the thing like Akash Tir has recently been in news like 3 or 4 or 5 days back. So we can leave those things, because these are very recent news and this year prelims will not cover them. So defense technology up to April, whatever technologies are in news, what DRDO has done in the recent past will cover all the news. Overall, if I may say, in the coming 2 to 2.5 hours, I'll try my best to cover approximately of 25 to 30 most important topics for this session. It may increase up to 40 as well, depends upon the time. So I will try my best to cover all these important news, starting with biotechnology. So when we are discussing about biotechnology first, or just a basic information that whatever PPT that you require, you can definitely go here, scan this QR code or search in the Telegram with the name of Shivam Sir PW. Definitely you will be getting the PDF of what we are discussing right now. So in this particular segment, let's start with the chapter biotechnology. And with the biotechnology, there comes a very basic question of DNA and RNA. I will say that DNA is the past news and RNA nowadays is becoming very, very famous with respect to genome editing or any type of biotechnology portion. This RNA is very much important this year. Earlier also, UPSC had asked several questions on the topic transcription. In this process, basically, our cellular system inside the nucleus, DNA gets converted into messenger RNA, mRNA that we commonly call it. So DNA to mRNA is definitely known as, the process is known as transcription process. But apart from that, there is one more type of DNA that was very common in news, and that is with the name of cell-free DNA. Cell-free DNA, what are these? Basically, we all know that in a structure of a cell, majority of the DNA or the genetic information carrier resides inside the nucleus. In this nucleus part, you will see chromosomes, chromatin fibers or DNA, whatever you may call, but all those genetic informations are inside the nucleus. There are certain extra chromosomal DNA as well. Extra chromosomal DNA denotes the DNAs that are not inside the nucleus, that are not inside the horizon of the nucleus will be termed as extra chromosomal DNA. For example, let's take an example of a organella which is inside the cytoplasm named as mitochondria. You all know that this mitochondria is having its own DNA known as mitochondrial DNA. These mitochondrial DNA will in turn make the mitochondria a semi-autonomous organella. Why is it so? Because this basic structure of DNA, the basic information that it entails, guides the cell. It is like the brain of the cell, where each and every information, where each and every genetic information is being processed and being stored in some type of nucleotide base pair sequences. So if mitochondria has its own DNA, then in somewhat nature we may call it a semi-autonomous organella, because it can function few of the parts from its own command rather than from the commands from the nucleus. So mitochondrial DNA is yet again an important topic which has to be covered today itself, not an issue. But let's move towards the extra chromosomal DNA, which we are calling the DNAs that are being apart from these things. So there are three basic terms. I would like to make differentiation between each of them. One is extra chromosomal DNA. Next come the cell-free DNA. And the third is environmental DNA. These three portions you have to pay attention here. Extra chromosomal DNA resides inside the cell and it is just hanging around in the cytoplasm. It may be residing inside the mitochondria part, or extra chromosomal DNA you can also see the similar thing in the bacteria part. Suppose this is a bacteria, these are the flagella part of the bacteria. This bacteria, since it is prokaryotic in nature, its genetic information is hanging around somewhere here. But along with that, it will also contain an extra chromosomal circular type of DNA, which is known as plasmid. So plasmid, what are plasmid? Plasmid are itself a structure of DNA, but it is under the segment of extra chromosomal DNA because it is not at all involved or it not at all counted itself inside the genetic information carrier. So these are the extra chromosomal DNA. Plasmids are the examples of it, and the mitochondrial DNA are the examples of it. Similarly, in the plant cell, inside the plasmid, you will see a similar type of plastid. So not plasmid, sorry, plastid. Inside the plastid, you will definitely see some kind of PT DNA, plastid DNA. So there are other extra chromosomal DNA. So this is one concept. The next one is cell-free DNA. Suppose when these cells get older, every cell has a certain lifetime, certain life period. After serving its whole life, it dies. When it dies, the structure disrupts. The structures, the structure that it held previously, it get distorted, or I may rather say that the complete cell membrane is widely open inside our blood streams or the fluid that is being flowing inside our body. So whenever we take a sample of our blood, these blood samples will also contain some of the cells that are not living in nature, or they have died. So these died cells have their chromosomes or the DNA segment floating in and around our fluid system. It may be blood, it may be other fluid as well, but it will be flowing inside our body as well. So whenever we take a sample of our blood, it will definitely contains the cell-free DNA as well. Cell-free DNA, as the name suggests, these DNAs are not bounded by the cell membrane. These are they already got free from the cell. That is why the name cell-free DNA. And the third one is environmental DNA. What are those? Environmental DNA. Whenever you see around yourself, suppose I am teaching at this point of time inside this room, my shells are shading. You might not see it, but my shells are shading at this particular moment as well. Not just mine, yours as well. So do not worry that nothing, you do not make yourself in difficulty that what happened to sir, the shells are shading. It is happening to you as well. Each and every moment we are shading cells. And suppose I am speaking, many of the drops of saliva may come out of my mouth and spread here and there. Same goes with the animals as well. Whenever an animal moves in a jungle, the hair may fall apart to the ground. The saliva, the excreta, or the blood sample, or the blood drops while they are running, any segment of their body which contains the genetic material can be collected. And through this environmental DNA, we can analyze that in this jungle, how many biodiversity, or we may say that how biodiverse is this region, how many floras and faunas are basically residing inside this. We may not see them in actual, but by sampling the DNA segment or the genetic materials that we have collected throughout this environment, we can get to know about them. So these DNAs which are spread around us, we may not see it. We might not, means our naked eye might not be able to see it, but there are plenty of information. The forensic science person definitely comes and visit a crime scene, pointing it out each and every details of it, and they collect very minute things as well, which can be considered as a DNA sample. So this is the environmental DNA. Now I see that you came to know about the cell-free DNA. That is what is being written here, that tiny fragments of genetic materials released into the bloodstream or other bodily fluids from dying cell. So suppose how we will use it. Earlier we say that cell-free DNAs are of no use, but nowadays cell-free DNA are of great use. Suppose a pregnant lady. For a pregnant lady, if we have to test the child inside his womb, and we might test that the child has Down syndrome or not. So earlier what we are doing is we are injecting a syringe inside the womb part and extracting the placenta of the fluid, amniotic fluid out of the placenta. And then we are figuring out that whether the child contains Down-free syndrome or not, because Down syndrome is a genetic disorder. So what we may say it, since the mother and the child is being connected with this umbilical cord, so all the nutrients are coming and going, means transporting in and out via this umbilical cord. So whenever we are getting a sample of the mother's blood, it will definitely contain the cell-free DNA of the mother itself, at the same time cell-free DNA of the child, which is being carried inside her womb. So by testing around these blood samples, definitely we come to know about the cell-free DNA of this child as well. And from those cell-free DNAs, we can arrange when we can test about the Down syndrome, whether the child possesses it or not. So these are the applications, these are the probable applications. Here it is all written. The next topic that is very, very important is the central dogma of molecular biology, where we see that a DNA is converted into mRNA, and then this mRNA goes to the ribosome, and in turn ribosome converts the information being carried by this messenger RNA into proteins. The process here is known as transcription, and the process here is known as translation. Many a time there is a reverse transcription. Where does it happen? When we see a virus with the genetic material as RNA. RNA are the single-stranded genetic information. Many viruses known as retrovirus possess genetic information as RNA. Whenever these retrovirus enters a host cell, what they do first? They try to combine their genetic information in the host cell's genetic information. But in order to do so, since the host organism's genetic information is double-stranded, means it is in DNA phase, while the virus itself has their genetic information in the form of single-stranded RNAs. So what they will do? They will convert these RNA into DNA. And the process of converting back the RNA into DNA is a reverse process. That is why it is known as reverse transcription. So reverse transcriptions usually happen when a virus, especially retrovirus, enters a host cell. The next very portion is microsatellite DNA. This is also an interesting topic about the DNA. These microsatellite DNA, suppose there is a full-fledged genome. The full-fledged genome contains 3.3 billion nucleotide base pairs. These 3.3 billion nucleotide base pairs does not contain healthy information in and around every nook and corner. Means there are segments of these nucleotide base pairs which are non-functional in nature. They are just there without any work, without any cause, without any information or meaningful information. So those particular base pairs which do not code for protein, which do not synthesize protein and are repeatedly in a way that they can be termed as short tandem repeats. These are the short tandem repeats. Very often they'll repeat themselves. And if these things are basically their patterns is traced, we found that earlier we thought that these segments of DNAs are of no use. But on a later date, we found that the pattern of their repetition is very unique for an individual, and it may serve as an identity for them or an individual. Means my STR's repetitions will be very much different from you all. So each and every individual possesses these microsatellite DNAs. The repeats are very different in each and every one of us. So for the forensic lab, for the DNA matching, for the inheritance, we often use the repetition pattern of these microsatellite DNA in the part of DNA fingerprinting. DNA fingerprinting is again an application of this, and we call it as polymorphic markers. Polymorphic markers because it may be used to study inheritance, or means to claim that who is the actual mother or father of that particular child, that is known as inheritance. And for DNA fingerprinting for the crime situation. Suppose someone has murdered someone and we have found some blood samples in the crime scene. We have few suspects, like 4 suspects, and blood sample. We'll collect DNA information of the 4 suspects and we'll match it to the blood sample that we found in the crime scene. Whosoever patterns is matched, since it is very individualistic in nature, definitely we will be getting our person, means who committed crime. So both of these things can be used here. Next, there is one more DNA which is known as extra chromosomal DNA, that is already been covered. Extra chromosomal DNA, it was in news because I'll just make a point. These are in news because they can also be treated these extra chromosomal DNA can also be treated as one of the cancer treatment. Maybe we found that whenever there is a tumor inside our body, we found that extra chromosomal DNA, the number of extra chromosomal DNA in and around that tumor cells increases. So we came to know that there is some type of role of these extra chromosomal DNA in the tumor section, and when we have inferred it, definitely we can control it, and by controlling the extra chromosomal DNA, there can be some solutions to treat cancer as well. So this is a probable kind of medicine which is being developed nowadays. We are still researching on it. The other topics of biotechnology which I think is important is under the name of genetic engineering. The next one is genome editing. Making the difference on these two basic headlines or basic topics will give you a lots and lots of understanding for this subject, for this topic. Genetic engineering, always remember that a genetic engineering will always involve a foreign DNA. A foreign DNA. This foreign DNA will be attached to some other species DNA. For example, suppose I am taking an example of E. coli bacteria. E. coli bacteria. The bacteria in itself contain plasmid, circular plasmids. Suppose there is a human being whose beta cells are not functioning at all, or we may say that beta cells, where we found it, beta cells are present inside our pancreas. What they do? They basically secrete, or they basically make insulin. And what does insulin do? These insulins, when we intake food, especially when we intake glucose or any carbohydrate, definitely our glucose level spikes inside our body. In order to maintain that glucose part and in order to send them to the most desired places of the cells, we need insulins. Insulin acts as a manager who manages the glucose level inside our body. So, if the beta cell or the insulin secretion of the beta cell is problematic in situation, or if it is not at all working, so we may say that our body will not regulate the glucose level, and we might find ourselves in difficulty when these glucose levels are not well addressed. So what we'll do? These patients are known as diabetic patients, and they often use external sources of insulin. They try to eat insulin, or they try to inject themselves with some insulin, so that their body can control the glucose level. So what we have done, where do we find this external source of glucose? We have first thought that the slaughtered pig and the pancreas, the slaughtered pig's pancreas will also secrete some of the insulins. We try to collect them, and definitely we try to make inside an injection, or we have converted into a pill to eat. So these were the earlier functions. But what we came to know about, that there is a bacteria known as E. coli, Escherichia coli. This E. coli bacteria, if we have our beta cell, suppose this is the beta cell. Inside the beta cell's nucleus, there will be a segment where we can find the what we may say, the information through which we can convert that particular segment into mRNA, and that mRNA goes to the ribosome, and the ribosome will in turn make this information into a protein which we call it insulin. So there is a segment inside the genetic information which codes for insulin. We try to take it out. We will take it out and we will insert the same piece of genetic information inside the plasmid of the Escherichia coli, E. coli. Suppose the genetic information which we gathered, we just fixed it inside the plasmid, and again we have placed the same thing inside a bacteria. Suppose this was the bacteria, and bacteria contains now a segment inside its plastid which code for insulin. So now this organisms, or the process that entails, is basically known as recombinant DNA technology. It is written like it, recombinant DNA technology. And under this technology, we have made a bacteria like Escherichia coli to secrete or to make insulin. This insulin or the segment of the gene that is being marked in yellow color is known as I gene part. You know that I gene is foreign DNA which has been inserted into the bacteria. Bacteria and humans are two different species and we have intermingled their genetic informations. So that is why this is being done in the genetic engineering part. And apart from genetic engineering examples of this insulin production, you can also take the examples of GM crops. So most of the GM crops, not all, but most of the GM crops basically known as transgenic GM crops. Always remember, not every GM crop is transgenic in nature, but the transgenic GM crops will definitely contain some of the foreign DNAs. For example, BT cotton. BT cotton is a genetically modified crop. You all know about it. So this BT cotton will definitely have some foreign DNA in it. Where this DNA is from? I would say that there is a bacteria known as Bacillus thuringiensis. That's why the name BT, Bacillus thuringiensis. It's a soil bacterium which secretes a toxin which is very harmful and it may cause death to the pink bollworm or the bollworms that are who were feeding on the cotton balls and making the cotton production in a bad situation, or we may say that they are hampering the cotton production. So what we did was, we have extracted that particular gene segment from the bacteria, the cry gene part, and we have transplanted the similar thing into the cotton plant. Now the cotton plant is producing the same type of toxin which that soil bacterium Bacillus thuringiensis was producing at that point of time. So now if a bollworm will come and try to eat cotton balls, it will eat and it will die. So in the similar segment, BT cotton also contains the part of foreign DNA. Same goes with the DMH11 as well. DMH11 is what? Dhara mustard hybrid 11. This was in news. Dhara mustard hybrid 11 possesses multiple genes like Bar gene, Barnese gene and Bar star gene. All these genes are being inside, all these genes are inside this DMH11. I will tell you an example if it is here, like, yes, this is the DMH11. The problem with mustard breed is like we have to make this mustard breed a VD site tolerant. What is the purpose why we are making this mustard GM? Genetically modified mustard is being needed for VD site tolerant. VD site tolerant is what? Whenever we are sowing the mustard in our fields, there are certain weeds that are being grown in and around the mustard, which sucks the nutrition which are for the mustard plant, but rendering the mustard plant with less nutrients. So in order to do so, what we try to do is to spray weedicide around them. Weedicide will definitely kill the weeds, but at the same time they will kill the mustard as well. In order to come out of this problematic situation, we came up with a solution that why not to make this mustard the weedicide tolerant, so that whenever we sprinkle or spray weedicide around in and around the field, the weed will definitely get killed, but the mustard will stand in their own way, means they are not even affected with the spray of weedicide for them. So weedicide tolerant will make them tolerant for weedicide, simple. So in this segment, the problem lies with the bisexual nature of mustard plant. So mustard plants are bisexual in nature, that is male and female. Both of these parts are already present inside the flower of a mustard. So they will not cross pollinate. And in order to have this, we have to insert these three genes which is responsible. One is responsible for herbicide tolerant, the other is responsible for making it cross pollination, for allowing the cross pollination, and the third is basically for the removal of male sterility, since this cross pollination will be done only in case if one breed is made to sterile in nature. Male sterility can be imposed, means for us, for our breed is Varuna breed, you can see it here, Varuna breed. We have introduced Barnese gene in order to remove the male part or in order to sterile the male part of it. Hence, the cross pollination from the European Hira breed can be done. And whenever the GM mustard is being produced, it also contained the Barnese gene, which is making the final product again sterile in the male part. So in order to cross it, in order to negate it, what we have done is introducing a one more gene of bar star gene in the Hera breed. What it will do is it will negate the male sterility. So finally, the GM mustard, what is being produced in here, will contain bar gene for the weedicide tolerant. The next is Barney's gene, which allows cross-pollination by sterilizing the male part of Varuna breed, and bar star gene is being introduced in the Hera European breed, which will remove the male sterility part in the offspring of these two crossbreeds. So DMH11 was made to approval for by environmental release by GEAC. But again, the commercial production is not yet being done. Always remember that commercial production in India is only for the BT cotton part, and no other crops is yet for the commercial production. So you get the idea of genetic engineering. Now comes the part of genome editing. What is this genome editing? Genome editing, acha, one more thing. In this particular segment, when we are saying that recombinant DNA, there is one more thing. What are the tools engaged in this? That tools what we are taking. So what I have told you is we have to cut apart a segment of DNA from some other species, and we have to join it or glue it to some other species. Definitely we will be needing something to cut apart, which will be known as restriction enzyme. These restriction enzyme will act like molecular scissor, and definitely there is ligase enzyme which will act like molecular glue. So molecular scissor and molecular glue in the form of restriction enzyme and ligase enzyme are being used in the genetic engineering. Now comes the next part of genome editing. When we say this as a genome editing, DMH11 we have already discussed. These things are already covered. Now genome editing or gene editing, both are same. Genome editing or gene editing is a part where we make a specific changes in the DNA. This can be done with the two prospective methods. The one is by knocking out some gene. Knocking out. What does it mean? Knocking out some gene means there is a set of DNA here, and we simply knock one particular part out of it. We cut this particular part, and these two parts will combine naturally. Combine again naturally. Means a particular segment which was faulty in nature, we have just removed it forever. This is gene knockout method, knocking out. The other is knockdown method. Always remember that when I was telling you that RNAs are now more important in here for this exam. So we have already learned that DNA is basically responsible for producing RNA, that is messenger RNA, and these messenger RNAs with the help of ribosomes are able to synthesize protein. So if we have the faulty DNA, then definitely it will produce faulty mRNA, and definitely in turn it will produce the faulty protein, which is not at all required for a particular organisms. Suppose in flowers or tomato used to develop a PG protein, polygaric urinase protein. So this PG protein is making tomato rot in like 1 or 2 days. So why to produce this? Why not to just make it silent? What we can do is we can remove that particular part so that we will never see the DNA part making the faulty mRNA or in turn the protein. The next part means one part is related with the DNA editing part. You get it? But once the DNA is converted into messenger RNA, means once the transcription process has already been done, then also there is a chance of editing in the RNA part. If even we are able to edit the RNA part, then also what we can do is we can definitely produce a good form of protein which was earlier faulty. The next thing is what we can do is, the third thing is, these are the two segments where we can edit the things. I am just explaining it to you. The third part is when this messenger RNA is reaching to the ribosome part, and then ribosome is making protein in the passage, in the way. If we are making something like that messenger RNA does not reach ribosome in any case, then also the faulty protein will not be produced. Means either we can edit the messenger RNA or we do not let these mRNA reach to ribosome. So these are the two other fundamentals that we can do with the messenger RNA part in order to edit the gene or in order to not make the protein. So this is one of the reason that knocking out a gene will always be a type where we cut apart a fragment of DNA forever. It may be for the DNA, it may be for the messenger RNA. If we cut apart a part of messenger RNA and cut it forever, means it will definitely a gene knockout method. But what we can do is we can have the gene knockdown method as well. That is what we are not interfering anything with the DNA. We always try to fluctuate the nucleotide base pairs of messenger RNA in two different ways. What are those ways? The first one is known as RNAi. RNAi. This is known as gene silencing method or RNA interference method. What it does? It basically you can see it here. Oh, sorry. I think it is not here with us. Not a problem. Not a problem. Where were we? Yes. Suppose what is gene silencing? Gene silencing. Listen to it very, very properly. Gene silencing. Gene silencing is a method which is also known as RNAi method. What we are doing is we are confusing the ribosome. Why we are confusing ribosome? Suppose there is a ribosome. This ribosome is getting an information from the messenger RNA. Messenger RNA. Whenever it is going to make protein, sorting out the information being provided by this messenger RNA, we are introducing one more part. We are actually making lab-made double-stranded RNAs. This is basically a double-stranded RNA. We call it dsRNA. So dsRNA in turn, it's a very complex process and it further gets converted into siRNA and rest of the things. But ultimately what it does, it will just create as a different segment of RNA with the exact opposite information or some information through which this ribosomes get confused that what to do next. Means it has both the information at the same time. The ribosome get confused that what to process, what is the actual recipe of the protein, and hence what it does is it just cut apart these RNAs of messenger RNA. These are the cleaved mRNA. Cleaved mRNA means ultimately these ribosome is responsible for making protein, but ultimately when it got confused, it did not create protein, rather it cut apart this messenger RNA. So ultimately the result what we were targeting is achieved, means the protein, the faulty protein is not produced. That's our purpose, and that is what the gene silencing method do. There is one more technology, what we call as this is microRNA. And why this microRNA is in news? Because in 2024, the Nobel Prize to Victor Ambros and Gary Ruvkun is for the discovery of microRNA and its role in post-transcriptional gene regulation. Now you know the meaning of transcription. Post-transcriptional means after the messenger RNA is being formed, and then we are regulating the gene part. They told about the messenger RNA. What is this messenger RNA? What is this microRNA? Suppose this is in the pink color, you see your genetic information carrier in the form of DNA. Once this DNA is getting converted into messenger RNA so that it will go to the ribosome and make protein, it also have a microRNA segment. Already it has. Always remember, always think like that. This mRNA is a chariot and the microRNA is like a sarthi. What it does, it guides the mRNA to reach to ribosome. If we confuse this microRNA, if we edit this microRNA in a way that this will never take this messenger RNA to the ribosome in order to make protein, then also we achieve our target, that is the protein synthesis will not happen. So in this particular segment, first of all, the Nobel Prize for this has been given for the discovery of this microRNA. That is, there are something called microRNA which guides this messenger RNA to the ribosome. This is point 1. And for the point 2, if we can edit this microRNA, then definitely we can make this messenger RNA not reach the ribosome. So in both of these cases, what we say this, Victor Ambros and Gary Ruvkun definitely got the Nobel Prize for this same segment. You can see it here, you can read it from here. The other part, what I am saying was the tools involved in these things. Suppose we can edit the DNA, we can edit the RNA, but just like the restriction enzyme and ligase enzyme as we saw in the genetic engineering part, what are the tools involved in this? The tools involved in here is like tools are CRISPR-Cas9. CRISPR-Cas9, where Cas9 is the Caesar, molecular Caesar. Next, similarly, we also get ZFNs, zinc finger nuclease. And next is talents. These 2-3 forms are basically made to edit the DNA part of it. Always remember, they'll just edit the DNA part, not the RNA part. In order to edit the RNA part, you need one more type of enzyme which is known as ADAR enzyme. ADAR enzyme. So you come to know that how the genome editing is basically working. So in genome editing, either you edit the DNA or you edit the RNA. If you want to edit the DNA, you need those tools. If you want to edit the RNA, you need ADAR enzyme. The next thing is, these are the gene knockout methods, but definitely gene knockdown method will also work. Knockdown methods, what they will do is, in knockdown, you are silencing a part of gene, means in order to silence the part of a gene, that is DNA, you are restricting it with the help of RNAi, that is interference, that is known as gene silencing, and the other is microRNA form. So these two will also work, that will change the way we look at this protein, or if this protein was faulty, definitely it will not be produced. So these are the methods through which we adopt genome editing. The third one is one more process that we call it as, and now there are few current affairs that is being related, that's why I have told you all these things, that is Casgevy and Lyfgenia. These are the two types of processes that is being approved by the US FDA, Food and Drug Authority, so that they can treat sickle cell anemia and beta thalassemia with the help of CRISPR-Cas9 tool. Earlier it was in news, earlier it was in theories, everywhere, but we have never used it on human beings. But for the very first time, the US FDA has approved Casgevy and Lyfgenia processes for the treatment of this sickle cell anemia and thalassemia. So that is why it was in news. And the other one is RNA editing, where I told you about the ADAR enzyme. The ADAR is nothing but the adenosine diaminase acting on RNA. This is the full form of ADAR enzyme. It is just working like the CRISPR-Cas9. Just in the DNA editing, we use CRISPR-Cas9, and just in the RNA editing, we use the ADAR enzyme. This is so. Now, the one more part of editing the gene comes in a way with the help of jumping genes. What are these jumping genes? Jumping genes are very natural in nature. In my body as well, in your body as well, there are plenty of jumping genes which try to change their places. They will jump from one location and they will settle them down in the other location of our genomes. And there might be some form like they have jumped, they have made their copies, and those copies will be transferred to other locations and then pasted. Means copy and pasting method will also be there, and this complete cut and paste will also be there, means deleting from here and producing it back here. But copy pasting will also be there, that one particular segment is being copied, it will be here as well, and it will be copied to other places as well. These are very natural in process, but recently the scientists have known about it, for the jumping genes. They thought, why not to convert these jumping genes into the way that they want to edit the gene? Means instead of getting these jumping genes in the natural way, why can't we moderate them? Why can't we handle them? We guide them. And some part of gene gets inactivated in a segment and activated in the other. This is the bridge recombinase mechanism, BRM. Bridge recombinase mechanism will definitely try to edit the DNA part with the help of jumping genes. So now you know that there is one more method of the jumping genes. The very next thing is gene therapy. When we use the word gene therapy, basically it tries to tell us that there in our genome segment there is somewhat a faulty gene is there, and we just have to replace this faulty gene with the healthier genes, so that the next time it will go for the synthesis of protein, definitely it works for the good protein synthesis. So if we are replacing this faulty gene into healthier genes, this is known as gene therapy. And again, this gene therapy can be of two types. One is of the germline gene therapy and other is of the somatic cell gene therapy. Germline gene therapy deals with the germ cells. Germ cells, it symbolizes that can germinate just like a sperm, egg, uh, embryo, zygote. These are the germ cells. And if other than these reproductive cells that are non-reproductive cells in nature, which is known as somatic cell gene therapy. So in this somatic cell, there are non-reproductive cells involved. And for example, what we can call it, here we can take an example of CAR T cell, chimeric antigen receptor T cells. And here you can take examples of MRT, mitochondrial replacement therapy. MRT is a very beautiful example. So MRT, we are engaging our egg cell or zygote in order to treat the mitochondrial disease. Suppose here you can see the CAR T cell therapy. In the somatic cell therapy, what we are doing is, suppose this is our T cell with some basic antigen receptors above itself. So what these antigen receptor do, they will definitely stick to the virus, or they will stick to the virus or any type of pathogen which in itself contains the part of antigen. These antigens will act like the gateway to enter the host cell. So if these T cells, if they block all these antigen with their antigen receptors, then the gateway will be closed and this virus will be of no use, or it will never reach to any host cell. So this is what a T cell, a regular T cell do. But many of the time when there is cancer cells are present inside our body, these cancer cells try to bluff these T cells. What they do, they ask T cells not to kill them because they are the integral part of the body. They will not cause any type of harm to this body, and T cell agrees to them. And that is why T cells never attack the cancer cells. But this is not the work of our immune system, means they are meant to attack them. In order to do so, in order to make them remember their power, we take out a blood sample. A blood sample will be taken out, and in this we try to segregate the T cells part, and on these T cells we try to modify it with the chimeric antigen receptors. We edit them with the help of gene therapy. What we do is we edit some type of we add chimeric antigen receptors to them. Now these chimeric antigen receptors are indeed very powerful, and they specifically target the cancer cells. And then when grown in the millions of numbers, we again infuse in our bloodstream, and again when this T cells reaches the cancer position or the body part affected with the cancer, definitely it starts killing those cancer cells, and over a period of time the cancer cell died. So this is a treatment of cancer. This is a treatment of cancer with the help of CAR T cell therapy. And why this was in news? Because for the very first time a company named NextCar19, for this NextCar19 has been approved by the Drugs Controller General of India, DCGI. So DCGI has recently approved this NextCar19 for the treatment of cancer in India itself for the very first time. That is why this CAR T cell was in news. The other is mitochondrial disease. So you have heard about the mitochondria, and you all know that mitochondria is the powerhouse of the cell. And whenever if a zygote is being formed with the faulty mitochondria, suppose this single zygote, we all a single cell once. When the sperm and egg of our parents fertilizes for the very first time, we all were a single cell named zygote. This zygote has been further developed into us, and we are now a trillion cells body. We have the trillions of cells inside our body. So if the very first cell, that is our zygote, if it contains some faulty mitochondria, you can think of the problems, because it is indeed the powerhouse of the cell. The trillions of the cells inside your body will be not functioning as per the energy requirement of the body. So definitely it might be like your brain would not have been developed as it is now. The body parts would not be developed like as it is now. So all such things will depend upon these mitochondria. And the very interesting thing is with the mitochondrial DNA part is that, suppose there is a zygote. I am drawing a zygote. This zygote contains some type of mitochondria along with a particular cell with the genetic material from the mother and from the father. Suppose pink color is for mother and purple color is for father. How it had happened? Because the mother has a eggshell with mitochondria, and inside the nucleus the mother has her own genetic material, and along with it there is a sperm from the father side, which also contain a nucleus part, and along with this nucleus there are certain genetic segments. So whenever these two fuses into a zygote, we always see that the complete set of zygote is being made from the eggshell. It is only the genetic part of the sperm which is being transferred to the zygote. So if a mother is having faulty mitochondria in her eggshell, then the same faulty mitochondria will be transferred to the zygote as well. So we all have got our mitochondria from our mother, and our mother has got her mitochondria from her mother, and then from her mother to her mother to her mother to her mother, and so on. We can definitely trace we can definitely trace this matrilineage. Maternal lineage can be traced with the help of mitochondrial DNA, because we all used to get these mitochondrial DNAs or mitochondria from our mother itself, because father, it is not like that father sperm does not contain the mitochondria part. Definitely it does as well, but since I told you that just the genetic information or the chromosomal 23 chromosomes from the father's sperm will travel to this and fertilize the egg. Rest of the part of sperm is of no use for a zygote. So always remember that mitochondria, if faulty, present inside the mother will be transferred to the child. And in this case, if this zygote is further developed into embryo and then into a person, in a living creature, then there is no treatment, because there is always the existence of faulty mitochondria in like trillions of cells, and inside a single cell we often see like thousands of mitochondria in one cell. So trillions of cells, each cells containing the thousands of faulty mitochondria, it is impossible for any treatment to take all of them out. So in this particular situation, what we do, we used to deal in the germline gene therapy. So what does this germline gene therapy does? It asks for a donor mother. So this donor mother have the healthy mitochondria with her, and along with that she had her in the, this is the donor's eggshell. This is the donor egg cell, and this donor egg cell will contain the healthy mitochondria. What we'll do, we'll replace this nucleus with the mother's nucleus in the egg part. So what we are doing is we are just taking this, just a second, I'll just make you very clear. And what we are doing is, if suppose we are taking out the nucleus from the actual mother with all the genetic information, and then fusing the same sperm with this, and the zygote formation that we are getting this time will be considered somewhere like this, that this is the final zygote, which contains the healthy mitochondria, which contains the genetic information from mother and father. So this is the zygote, this is the healthy zygote, and when it will develop into a person, definitely the person will be healthier in nature. So this whole process is known as three parent baby, And three parent baby is always because it involves the genetic information from the three parents. One is definitely the mother and father, and since this mitochondria also contains DNA segment, it has the DNA part inside it. So three parent baby, three persons DNA is being involved in this zygote formation. So always remember that this mitochondrial disease or the treatment of MRT, mitochondrial replacement therapy, can happen at both of the ends, that is before the MRT takes place, that is before the fertilization the MRT takes place, and even after the fertilization. For this as well, even if we have zygote like this, then definitely we can ask for a donor zygote and we will replace this zygote nucleus into that particular segment, and again we will be getting the same zygote. So this is the reason when we have seen like MRT has two processes. One is spindle fiber transfer and the other is pronuclear transfer. This one what we have seen is the spindle fiber transfer, and again where the zygote exchange takes place, definitely it is known as the pronuclear transfer. The other one more thing is stem cell therapy, and here as well the stem cell therapy can be one of the type of somatic cell therapy. Somatic cell therapy, how it works? Stem cells. What are stem cells? Stem cells are any type of cells which have the ability to regenerate into other types of cells indefinitely. What does it mean? That suppose I have a zygote. This zygote has power to develop itself into any type of cells like brain cells, stomach cells, liver cells, bones, muscles, cartilage, any type of cells the zygote is capable, and that is why it can be converted into any form of the cells, and hence the name totipotency. Mean the potential of this particular zygote is total in nature. It can be converted into any type, and that is why totipotent cell or stem cell can be considered for this. Zygote is an example of totipotent stem cell. And as we move ahead, definitely for the embryo part is like pluripotent, where the potency has been decreased a little bit, where it cannot produce embryonic stem cells or embryonic cells like placenta, umbilical cord, and so and so. The other one is multipotent, and the next one is oligopotent and unipotent. So each and everything is considered under the potency of into how many cells they can regenerate. For example, there is a cell known as mesenchymal stem cells in the blood of in the period blood of a woman. So these mesenchymal cells are multipotent in nature because these mesenchymal cells can regenerate themselves into bones, cartilages, and muscles or tissues. So that is why they are known as multipotent. Again, we have like hematopoietic stem cells. Hematopoietic stem cells exist in our bone marrow. In the bone marrow part where the bones are being produced, these hematopoietic stem cells can produce B cells or even before that RBCs, WBCs, platelets. These WBCs can be further of like various types like neutrophils, like phagocyte, not phagocytes, neutrophils, and what was the other one? Neutrophils and lymphocytes, like lymphocytes. These lymphocytes can again be developed into T cells and B cells and so on. There are multiple stages. And unipotent is like our skin. Our skin gets only regenerated into some other skin type. So these are unipotent in nature. Now you get to know about all those things. So what we are doing is we are using a pluripotent stem cell, where we can get this pluripotent stem cell. Suppose a child is being brought into this universe. At the time of birth, when the umbilical cord, when the mother's body and the child's body is being connected with the umbilical cord, you often have seen, you might have seen, I think you might have seen that umbilical cord is there. So while getting this umbilical cord cut, and a piece of umbilical cord can be taken as the stem cell for the pluripotent stem cells. In the future, we often see that currently, although we do not have the technology to regenerate these stem cells into some type of organs or tissues, but in future definitely we would be having this technology. So in order to do so, pluripotent stem cells are being preserved at the stem cell banks, and those stem cell banks used to keep these stem cells of a particular individual or a child for next 20 to 25 years in any such situation if the child faces a tragedy, so that these stem cells can be used in order to build some type of organs in the future. So this is what stem cell therapy means. The other thing is genome sequencing and genome mapping. Suppose what is genome sequencing? Genome sequencing is a very common term. Suppose we have 3.3 billion base pairs, and we just we just sit down and note each and every sequences, like the first one is A, then T, then C, then G, okay, then A, then C, then C, then T. And we sequence of like 3.3 billion nucleotide base pairs. This will be known as genome sequencing. It is time taking and very costly in nature. But in order to have it very quick, we often refer to the genome mapping part. Genome mapping is not the complete overview of the each nucleotide base pairs, rather it is just a particular structure. This is a general overview. This general overview, what it does, it will just create a map of an organism genome. We will first set out a location for which we have to oversee it in a general way, and for that particular location we'll try to find out the problem. Suppose someone has a cancer cell, or suppose the CAR T cell. So what we know is we can definitely pull out some WBCs, and in WBCs the genomes of a particular segment is judged, and in order to get that we just go for genome mapping, not the complete set of genome sequencing. So it is easier to go for it, it is cheaper in nature, it is quicker in nature. Other thing that is very common in the news for this year was Bio E3 policy, which corresponds to biotechnology for economy, environment and employment. It basically goes for creating or establishment of these three things. That is, one, biomanufacturing facilities. What are they? They are basically like, as I told you recently, that Escherichia coli, E. coli is used to make insulin. So if we are mass producing this insulin in a lab scenario or in a manufacturing setup, then that particular biological setup will be known as biomanufacturing facility. The other is biofoundries. Biofoundries always incorporate some type of advanced tools and technologies for the research and development part. Means there must be something very new which has not been done yet, and we are researching on a very new things of like new type of medicine, just like organ on chip can be a thing. I'll just tell you that what is this. But these are the biofoundries. Inside these biofoundries, we will be researching and developing new forms of medicines. And BioAI hubs is a combination of biotechnology and artificial intelligence, where a complete set of data from the biotechnology part will be given to the artificial intelligence in order to stretch some of the patterns and some of the new form of medicine or the new research part. This is the function of BioAI hubs. Now, we came to know about some of the new types of medicines in today's world. These are just not the pills of paracetamol or the injections or the vaccines, but it's completely a different approach of these 3D cultural models. The first one that we often see here is 3D spheroid organoid bioprinting and organ on chips. 3D spheroids are just the computer part of it. These computer algorithms will create it digitally, and we can test all the medicines on it. The next one is organoids. This is particularly the brain organoid part, where the brain organoid part is injected with lots and lots of electrodes. It is just work like the artificial neural network, which is going to be discussed in this lecture at the same time. The next one is bioprinting part. Bioprinting is a future technology. With the help of stem cell therapy, we can print few of them. Organ on chip. This is a 1.4 billion market by 2032, where we are basically using the particular organ cells inside this, and for the testing of drugs we are not using any particular animal, or the animal testing will not be there. So in this way what we are doing is we are having this organ on chip. You can see it from here as well. That organ on chip is an integrating cells derived from a human body with a well-defined in vitro biological environment. So what it will do, its major purpose is to replace the use of animals to test drugs. The next part is biosimilars. Biosimilars are often in news. You have also heard about the generics part, or you have heard about yes, generic medicine. So what are these generic medicine? Suppose there is a company known as Cipla. It is researching on a medicine for long 10 to 15 years, and after that they came to know about a solution, a complex solution which will be able to treat some type of disease. So now this chemical composition, if we give the chemical composition, the information of this chemical composition to a third company, saying that this is the particular amount and this is the particular recipe to produce this tablet, just produce it. So the third company will, they have never spent any amount in the research and technology, and definitely although the manufacturing cost for that one tablet is 0.5 paisa or just the 5 paisa or 50 paisa, the Cipla company will be selling it on a price of like 100 or 200 rupees, just because they have to uh, inculcate the amount invested in 10 to 15 years of the research. But for the third party company, which has not been involved in any type of research or any other thing, what they did, they will produce it in the way like 50 paisa and they will sell it in 60 paisa or 70 paisa. So these generic medicines, we in India see a lots and lots of opportunity in generic medicine, and we often export a huge amount of medicines to the third world countries and also the developed countries like USA. But these are the generics, these are the generic medicine. Then what is biosimilars? Biosimilars is somewhat different in a similar way. Suppose take the example, again I am taking the example of insulin production from the E. coli bacteria. This is the process. There is no chemical composition of this insulin. There is a process, there is life involved, there is bio involved in this. And when this bio or life involved in producing a type of medicine, and we are replicating the similar type of complete set of process, we call it biosimilars, not the generics, because it involves the living part. Suppose Escherichia coli, E. coli, you can get it from here. That biosimilars requires extensive studies, and definitely the process cannot be completely copied, because again the life acts differently in different time. So again there is a requirement of clinical performance, or there is a requirement of like some I may say it like approval. Yes, approval is required for the biosimilars, but for the generics, as we know that chemical composition is same, the production process is very similar. It means the same medicine will be effective in the same manner that the Cipla company has produced earlier. So it does not require that particular approval, further approval. But biosimilar will definitely require further approval, since the process cannot be copied in the same manner. So how this has been made and how effective this is in the new scenario, we have to test it again. So the manufacturing process is literally very complex in here. Here the manufacturing process is too simple. We just have to know the chemical compositions and just we have to make it by mixing them. But here the process is very different, highly similar to an approved biologic. These biologic, what are these biologic? You can say the insulin produced out of that particular E. coli is a biologic. So made from living organism, there is no such chemical composition involved in here. A minor difference in the process can make some different product. Instead of insulin, we will be making some other products by varying in the variations of very small things, we say. So the next thing is heritable human genome editing. This is just one thing. When the word heritable is here, heritable is something which involves germ cells. So heritable human genome editing is nothing but germ cell gene therapy, just like mitochondrial replacement therapy. Also know that mitochondrial replacement therapy is in news because there is a country in Africa and one more country is there. These are just two countries which has allowed germline gene therapy. Otherwise, in rest of the countries, it has been barred in the grounds of ethical dilemmas. Like it might be possible that we will be able to produce designer baby while changing few of the genes inside the germ cells. So while playing with the germ cells, we can produce designer baby, and again it will increase the social distance, social differences in the upper class and the lower class. in a way, like the upper class who had the money, their child will be definitely beautiful in nature, more mindful in nature. But for the poor, it will not so. It will not be so. So definitely the designer baby problem is there with the same germline gene therapy and along with heritable human genome engineering. So the next topic is health and diseases. Health and diseases, in that particular segment, the very first topic is related with the vaccine formation. We often see that even after the COVID, where we have seen the plenty other methods of producing vaccines, in which the mRNA vaccines are the most common and the most complex one, we see that there are other types of vaccines that are live attenuated, inactivated vaccine. What are they? How our immune system works, we have to make this clear first of all. So, how our immune system works? Suppose we have already been given vaccine to it, or we are innately, means from the very birth, we have already immune to a certain type of virus or a pathogen. We derive it from our parents. So, from the birth, we ourselves are immune from certain diseases and pathogens. Suppose those pathogens, if they enter inside our body, our immune system definitely responds by providing or by making antibodies. In that particular segment, since the recipe of making these antibodies were already present inside the genetic information of our immune system, these antibodies will be produced instantly. But suppose there is a situation where we are not immune to a particular pathogen and a very new kind of pathogen enters our body. The immune system then also can make antibodies, but it will take time now. Take the example of corona. COVID virus, the coronavirus, when enters into our body, it entered it for the very first time and our body encountered it for the very first time. So it could not understand the formation and it start working against that particular pathogen. But it may take 10 to 12 days. For that certain period, 10 to 12 days is more than sufficient for that virus to multiply itself in various host cells. So sometime it may cause death, means, and sometimes it will give you the nausea feeling or the headache or the fever, or um, the very common thing that we often see in the COVID was the loss of energy inside our body. So in that particular time, we went for the vaccination. What these vaccines do? We often use vaccines in the form of virus itself. We were using coronavirus either in the inactivated form or died form, or in some way which is itself is live attenuated but in its weaker form, means it will not impact our immune system of our body, or it will not cause harm to our body. In that weakened form of virus or dead form of virus is used. So whenever we are injected by dead form of virus or weakened form of virus, again the trigger of our immune system will act, and this trigger will again be taking around 10 to 12 days in order to make antibodies for that particular weakened form of virus. Because for our immune system, it is means they are not knowing that this is weakened form or these are the dead form of virus. For them it is a pathogen, and for that pathogen they have to produce antibodies. And once these antibodies are produced, our immune system saves a portion of information or the recipe of making these antibodies in our genetic information. So we know that how the next time if the actual virus comes, then what we have to do, what our immune system have to do. When we face some real COVID virus for the next time, our immune system immediately remembers it and start producing antibodies, because it has already produced it in its weakened form or in the dead form of virus. So these are the two forms of vaccines that we usually give. The other is subunit vaccine. Subunit vaccine, why it is important? This is so because we came to know in the later stage that our immune system does not recognize the complete pathogen. They just remember the antigen part of it. What are the antigen? You have often seen a virus with some spike proteins around it. Even earlier when I was explaining the CAR T cell, even the cancer cell that I have shown you with some certain antigens. For that, our T cells have Y-shaped antigen receptors. You get it? So, these different types of antigens will have different types of antigen receptors. So, in that particular thing, our antibodies, whatever these Y-shaped antibodies are produced, for each and every pathogen, these antigens are different and our body remembers those antigens only. So we thought that why to give dead form of virus or weakened form of virus in the complete set of a virus? Why cannot we just inject this spike protein or the antigen part of it or any part of it which can trigger our immune system in a way that it may produce the antibodies against it in within the 10 to 12 days, and since the genetic information has not been there inside our body, the virus will not harm in any possible way. So these are known as subunit or recombinant vaccine. Subunit is a different form otherwise. Recombinant vaccine is a recombinant DNA part, where what we are doing is we are adding the segment of DNA which is being responsible for producing the spike protein inside the pathogen. We are taking out that particular part of genome from the bacteria or virus or the pathogen, and we are transferring it in some other form of virus which are not that harmful to us. Suppose any type of adenovirus. So that is recombinant vaccine. Subunit vaccine is using any segment of the virus which can trigger our immune system, and next time whenever the actual virus will come, it will remember the same and act accordingly. So subunit vaccine has an example of messenger RNA. Messenger RNA vaccine. What we are talking about is not just getting out the DNA part, genome part of it. We are producing messenger RNAs inside our lab. Suppose for the COVID virus, some great scientists have sequenced its genome, and genome sequencing can be transferred through the data part, means we can transfer it on the internet part, no issues with that, it's just a data file. So it can be spreaded, it was open source as well, anyone can download it, and by downloading it, anyone can know that particular segment which codes for spike protein. We convert them into the messenger RNAs in the lab, and these messenger RNAs, when enclosed into some nanoparticles, were sent inside our body, and that particular body will enter into the cell, go to ribosome and make a spike protein. We are wasting like 5 to 10 years in the research part just to make those protein inside the lab, but we forgot that our body has the innate ability to produce protein from the ribosomes. Ribosomes have the innate ability to produce proteins. So whenever it has been provided with messenger RNA, definitely it will code for protein. It will code for the protein or any information which is being carried by the messenger RNA. That is why we have taken the messenger RNA part from the virus itself, and that particular messenger RNA will code for the spike protein. We definitely have encoded or we have wrapped it inside. These messenger RNAs are wrapped inside a nanoparticle, you say. Nanoparticles are basically used so that they can enter into the cell. So when they can enter into the cell, these messenger RNAs go to the ribosome, and definitely they with the process of translation they produce the spike protein, and our immune system definitely triggers it. So these are the various things that we have already discussed. So there is another known as HPV vaccine. HPV vaccine is basically for the cervical cancer, which is being very common in the women of India or in any particular country. But in India, the cervical cancer is very common. We may say that in every year there are 75,000 deaths approximately for this cervical cancer. So what it does, cervical cancer, what it is, there is a virus which is known as human papilloma virus, HPV. This HPV virus basically affects the genital parts of the women, basically cervix or and causing genital warts in the cervix, anus or throat. So in the particular area, the women are mostly affected by this cervical cancers. So what we are doing is, inside our immunization program, we are giving free vaccinations against this HPV virus, human papilloma virus, and in the 3 phases, for our child girls in the age of 9 to 14, they have been provided with this immunization program, with the vaccines, so that in the future they will not be able, they will not be getting this cervical cancer. So who produced this HPV vaccine? The Serum Institute of India, the same company which produced Covishield in the time of coronavirus. So the next is Havisure hepatitis A vaccine. It is also in news. It is the India's first indigenously developed hepatitis A vaccine virus, which is under the name of Havisure. What are these hepatitis? Suppose there are 4 or 5 types of these hepatitis, where the extreme part, hepatitis A and hepatitis E, the extreme segments of both of these corners, A and E, they are basically have something in common, that they are basically being produced while we are not maintaining good hygiene or sanitation. So by drinking means not safe water, means how we can prevent it? If we are drinking contaminated water or food, or we are not maintaining the proper hygiene and sanitation, the hepatitis A and E can happen. So both of these can having the virus, hepatitis A virus and hepatitis E virus. But for the rest of the B, C and D, B, C and D is having the transmission level through blood, through sexual contact, through mother to child. So hepatitis B and C and D, these three things, we can say the symptoms is like fatigue, jaundice, abdominal pain, joint pain. These the symptoms will be same, but the prevention can be done from the safe sex practice and sharing needles and avoiding sharing needles. So yeah, so in this segment there is one more thing that we have to take in our remembrance, is that hepatitis B is the only virus with DNA. Means hepatitis B is the adenovirus, and rest A, C, D and E are the retrovirus. There is one more thing, that for the hepatitis C there is no vaccines available till date. For A it is available, for B it is available, for D the same vaccine which were being used for hepatitis B is used for hepatitis D, and hepatitis E has also the vaccine available in it. The next is global TB report that is being published by WHO in 2024. So in this particular segment, we say that we have missed our targets. The target was like from 2015 to 2023, we have to reduce the cases of TB by 50% and death by TB by 75%, but we have just achieved 17.7 and 24% respectively. But apart from it, there are certain other means report in this particular information as well. The particular information in this, that 25.1 lakh in 2023, India saw an increase in reporting cases, which is good for us, that we are actually working on this TB, and there is a slight decline in the cases and death. Very slight, we can say that it was like 3.31 lakh in 2022, it goes down to 3.2 lakh in 2023. So a very slight change, I would say. India has the goal of eliminating TB by 2025, earlier than what the WHO prescribed till the date of 2030. But as we are seeing here that it is a bit difficult for us to achieve, but again we are trying our best. There is one more thing that we have to know about the TB is, there is a simple TB, all what we can say as pulmonary TB, and there are some extra pulmonary TB which affects the other part, like gastrointestinal TB, skeleton TB, liver, etc. But there are something for the treatment as well. If you go to any doctor, definitely they'll prescribe these 3 medicines like rifampicin, isoniazid and pyrazinamide. Isoniazid, rifampicin and pyrazinamide. These 3 medicines will be given to you on the very first moment, and it will go for like 6 months or 8 months straightforward. But suppose a patient visits and we come to know that these 3 medicines or itself the isoniazid and rifampicin is not working on that particular patient, we call that patient with a TB of multi-drug resistant TB. He or she will be having multi-drug resistant MDR TB. In this segment, isoniazid and rifampicin will not be working for his body. We will use the second line of treatment, that is some kind of injections like amikacin or kanamycin some capreomycin. These medicines with the injection form is treated as a second line of medicine. But even if this isoniazid and rifampicin is not working, we understand. But for some patients, this second line of drugs will also not work. At this point of time, the TB becomes the XDR TB. And if any type of the medicines, even the after medicines, suppose if a patient is diagnosed with MDR or XDR, what we'll do? We'll give them some other line of treatment. The new treatment that India has adopted is the BPAM regimen. BPAM regimen. I'll just give you the details of BPAM as well, but always remember that if a patient is having MDR TB or XDR TB, he or she will be given BPAM medicine regimen. BPAM regimen will be provided to him. But even if the BPAM will not work, definitely nothing, not at all any medicine is working on him or her, then it is a case of totally drug resistant tuberculosis, TB or TB. So what is BPAM regimen? BPAM regimen is the new part of drugs. It contains 4 drugs: bedaquiline, pritaminide, linezolid or moxifloxacin. These 4 medicines are there, and it has been treated under the National Tuberculosis Elimination Program. It has already been there. And earlier we say that whenever there is when there is a patient of MDR and XDR TB, it will usually take 20 months of the treatment. But with the BPAM regimen, it is hardly getting 6 months. 6 months of treatment and the TB can be eliminated. There are certain I can say programs running by the government. There are certain yojanas. You can say Nikshay Poshan Yojana. Earlier Nikshay Poshan Yojana was one of the yojana where we say that 500 rupees per month will be given to any TB patients from the any economic background. He or she will be given 500 rupees per month. But now Nikshay Poshan Yojana by the Union Health Ministry, the amount of 500 has been increased from 500 to 1000 rupees. So this is a centrally sponsored scheme under the National Health Mission, and the Ministry of Health and Family Welfare is the body who has launched it. Basic aim is to provide the certain nutrition for the TB patients. So by this 1000 rupees, they will be able to get their nutritious food. So other type of vaccine is mRNA vaccine, which we have already discussed. Yes, there is a second dengue vaccine, TAC 003 live attenuated vaccine, not that much important. The again Serum Institute of India has also produced the R21 Matrix M malaria vaccine. For the malaria also we have the vaccine now. The next thing is NOMA. This is important. This is regarding the neglected tropical disease. What are these neglected tropical disease? These are a group of conditions which are being prevalent in the tropical regions and being neglected by rest of the countries, and there are certain stigma attached to them, and means they can be caused by various of pathogens. There is no boundary for that. They may be caused by viruses, bacteria, parasites, fungi and other toxins as well, but they are referred neglected because they do not find their place in any global health agenda, or they are getting their low global fundings, and definitely there is a part of a stigma and social exclusion attached with them. But in India is having like 10 types of these entities, which is the highest in the world. World's largest absolute burden of at least 10 major entities. You can say that WHO has recently certified that India is free from guinea worm disease and yaws in 2000 and 2016. And again, WHO has told that India is being freed from trachoma as well. So WHO has recently said that trachoma, a kind of problem is with our eyes, where our eyes got a disease in a way that gradually the person will become blind after getting this trachoma disease. So definitely the sanitation, another thing was the basic reason why the trachoma is happening, because of the lack of sanitation and the poor hygiene and the poor nutrition, and it is basically affecting the child of the age 0 to 5. Trachoma, you can see it from here that recently the WHO had declared that the government of India has eliminated trachoma as the public health problem. So earlier also what India has eliminated is smallpox, polio and trachoma. Trachoma recently it has been added. So there is one more disease which was in news under NTD is NOMA. NOMA is basically also known as Cancerum oris or gangrenous stomatitis, where in NOMA is like in poor countries with the child aging 3 to 10 with I say poor oral hygiene or malnutrition or weakened immune system, they are getting this Noma disease as well. So again, there is one more thing, Wolbachia method, where we are infecting these mosquitoes with some type of bacteria known as Wolbachia. They already inherit them, but we inject these Wolbachia bacteria into the male or female mosquito in a way that if any of them is not having the Wolbachia injected, the offspring produced will not carry Wolbachia. But if the male person imbibes Wolbachia, there is no eggs will be hatched or no offspring will be produced. And in both of these cases, whether either the female is Wolbachia enabled or both of them are Wolbachia enabled, the offspring will be produced, but the offspring will itself will be Wolbachia enabled. When the Wolbachia enabled offspring is produced, they will not be able to become a carrier of a disease or a vector of a disease. So this is how we are eliminating the problems related with the dengue and rest of the things. The next one is traditional medicine. For the very first time, WHO has signed with the Ayush Ministry to open one and only WHO GTMC, that is Global Traditional Medicine Center. This is just one in the entire world in the Jamnagar, Gujarat. What it will do, it will spread the awareness about the Ayush thing, that is Ayurveda, Yoga, Siddha, Sovarikpa, Unani and homeopathy. The other one in news is H5N1 bird flu virus. These avian influenza, which we call is H5N1, this H and N is basically we can say it is categorized by some of the proteins. These H and N are the proteins of hemagglutinin and neuraminidase. These are the proteins, and hemagglutinin is of 18 types and neuraminidase is of 11 types, of which it is H5N1, which contains that fifth version of hemagglutinin and first version of neuraminidase is present inside this avian influenza. The transmission can definitely from the bird to human, but for human to human transmission, this is still one mutation behind, so that even we can say that after a while, means after 3 or 4 or 5 or 10 or years, when this mutation will happen, definitely human to human transfer of avian influenza will also happen. Next, mortality rate is too high in that particular segment. The next portion is space technology. Space technology is basically space technology. In this space technology, the very first thing is the stellar cycle. Stellar cycle is basically a form where we see that a low mass star will usually be getting older, means it has a life much more than a massive star, because the fuel which is being used inside it will burn at a very slow rate, and it will be used in very long period, making it a longer life for this low mass star. It will be ultimately converting into white dwarf, and for this massive star, it may either be converted into neutron star or a pulsar or into a black hole. This all will be dependent upon the core mass that it contains at its last stage. This core mass is being judged by the Chandrasekhar limit. We say it Chandrasekhar limit, which say that for our sun, suppose our sun's mass is Sm, solar mass is Sm. This core mass, if it increase, if it is more than 1.44 times of our solar mass, then definitely it will be converted into neutron star. If this core mass is less than 1.44 times of the solar mass, then definitely it will be converted into white dwarf. And if this core mass is more than 1.44 times of the solar mass, but this neutron star will also happen only in a case if this core mass is less than 3 solar masses, and if the this core mass is more than the 3 solar mass, then it will convert into black hole. This is what 1.44 times defines, and this is the Chandrasekhar limit given by this Subramaniam Chandrasekhar, the famous scientist from the India. There are other types of star, like Cepheids is one of the most brightest star in the universe. It periodically dims and brighten. It is periodic in nature in a way that we may even fix our clock with the periodic nature of dimming and brightening. The other one is pulsar. When neutron stars get fast rotating, emitting a stream of lights from the various ends. They will be known as pulsar. Next is quasars. Quasars are basically a form of black holes or a supermassive black holes emitting a lot of light. You can see it from here. Very often we see in the center of the galaxy with these supermassive black holes, and these supermassive black holes surrounding them in the accretion disk part, there is a huge amount of light which can be seen from our naked eyes even from here, but since they are very far from us, we cannot see them, or the effect of these very bright light is not affecting on Earth. The next one is James Webb Space Telescope. It has recently seen a brown dwarf. What are these brown dwarf? Whenever we are finding the white dwarf at the death of the star, or a low mass star, death of the low mass star, once formed the white dwarf, and the light is still in there, but when the light vents out in this white dwarf, then it gets converted into black dwarf. Black dwarf is completely conceptual in nature, since our universe is not that much old. That is why we can say that black dwarf is not at all present in our universe. But what is brown dwarf? Brown dwarf are basically the failed star. They are not able to convert themselves during the nebula or the dust and cloud part. Dusty cloud part will not be able to make themselves into a stable star. Those are known as brown dwarf. They never made themselves into a star. They are the failed star. There is one more concept with the term gravitational lensing and the Einstein ring. What it says that whenever you are using a telescope to view a galaxy, view a distant galaxy, but in that particular distant galaxy, there is one more galaxy which is being held in between these two things. So what it will be doing is, this galaxy will be distorting the space-time in a way that the light coming out from this distant galaxy will be bending towards this. Each and every light will bend, and not just with the up and down, the front part and even the back part will be bulged for this, we may say that, this galaxy. So space-time graph will be curved, and hence the light will be curved, and for us the light, what it will make, the image it will make is something like a ring like a structure, and this ring is known as Einstein ring. And definitely this is particularly the Einstein ring, along with the phenomena through which this galaxy is acting like a lens, just like a biconvex lens. That is why this phenomena is known as gravitational lensing. There is one more famous telescope, that is Major Atmospheric Cherenkov Experiment Telescope, MACE telescope. It is the state-of-the-art ground-based gamma ray space telescope, which is being inaugurated in the Hanle Ladakh part. Hanle Ladakh part in the Ladakh region. So what is this Cherenkov telescope, or what is this Cherenkov effect means? You can clearly see it from here. If suppose there is any particle which travels faster than light in any particular medium, yes, it may happen, because suppose there is water. In this water, suppose the light particles or the photons are traveling with a speed of like 75% of their speed which they possess in the vacuum. So in the vacuum, the speed of light is fixed as c, which is 3 lakh km per second, but in water definitely this speed decreases. And in this particular moment, if there is any other particle other than this light which can travel faster than light, then it will definitely show the Cherenkov effect. In this Cherenkov effect, when this particle is traveling faster than the speed of light in this particular medium, it will strike the atom's surface, and it will strike electrons out of these atoms, making them glow in the blue light. These blue lights will definitely be termed as Cherenkov effect. There is one more news very recent, that the Italian scientist has frozen the light. What they did, how UPSC will ask questions on it. The very first thing, that what is being frozen? This is not light which is being frozen, rather it is polariton. What are polaritons? Polaritons are the hybrid particles. They are not just photons, but along with the photons, we have coupled them with excitons. Excitons, what are excitons? They are simply a pair of electron and holes. This whole combined coupling of photons and excitons termed as polaritons. So we have not frozen light, instead we have frozen polaritons, which indeed works or acts like light in here. The next thing is, what is the material in which we have trapped the light? Suppose there is a speed of light at 3 lakh km per second. We are making such mediums where the speed of light tends on decreasing and decrease till 17 meter per second. Just 17 meter per second. From 3 lakh kilometer per second to 17 meter per second, we have created a different state of matter which we call it as super solid state. In this super solid state, you will not be able to imagine it, but it is like super solid like rigidity. Means if you will hit it, you will be getting pain in your hand, because this is solid like rigidity, but at the same time it will be super fluid like flow. Means without any friction it will flow as well. So it is very difficult to imagine that how these state of material will look like, but this is known as super solid state. Apart from this super solid state, we have definitely solid, liquid, gas, plasma, Bose-Einstein condensate, and many others state of matter. So there are not just 5 or 6 states of matter, there are plenty, and among them super solid state is one. So super solid state of matter is definitely working as a medium in which the light is being trapped. So what we trapped is polarity, and in which medium? Super solid state is the medium. So it will be definitely used in the quantum mechanics. The other one is related with the space technology is the orbits. So for the orbits, there are certain orbits like low Earth orbit, medium Earth orbit, geosynchronous orbit, and high Earth orbit, for which we have various types of satellites which are placed in each and every mediums. There are certain polar orbit as well. Suppose we have remote sensing satellites, so polar orbit are the best. If we have to observe the Earth, then low Earth orbit is the best. If we have to communicate or we have to fix the satellite on a particular point, then geosynchronous orbit or the geostationary orbit is the best orbit. Or if we have to launch some global type of navigation satellite, then medium Earth orbit is the best. So if each and every orbit is basically having its own function, there is one more orbit which is known as halo orbit, and these halo orbit are basically on the Lagrange points L1 and L2. This L1 and L2, since the name halo orbit, it will revolve around the sun, but with respect to Earth as well. So these bodies will always be in between Earth and sun. It will move along with the Earth in order to make in L1 it will continuously look towards the sun, an uninterrupted view of the sun, and for the L2 it will have the uninterrupted view of the space. So these both of these points, the objects placed in it will be traveling in the halo orbit. Next one is the privatization of space sector, and for that ISRO has already been working since past 1992. First of all, it went for an Antariksh Corporation Limited, which acted as a commercial wing of ISRO, and it is still acting as a commercial wing of the ISRO in the foreign market, for telling that what ISRO has produced, manufactured, and if the technical consultancy is required from the ISRO part, all is chargeable, and definitely we can seek consultancy services as well from the ISRO. So in 2019, NSIL is being made in order to make the same commercial arm in the national market as well, and NSIL is being given responsibility of producing and launching so many of the satellites and launch vehicles as well. Even the SSLV part, which we will be learning just now, is under the NSIL part. Next comes the in space. In space, as the name suggests, it is the basically authorization center. This authorization center is basically made in order to suppose you have to establish some kind of industry clusters, or some companies need some type of establishment and authorization, or any type of you can say permission to do something in space will be dealt with this in space. Next one is ESPA. ESPA is not the governmental body, means whatever you have seen here, all these three bodies worked under the Department of Space, and apart from ISRO, since ISRO is single-handedly dealing in research and development, these three organizations act like a private body. And the other is in 2023, May 2023, the new space policy is announced the same. It announced the same thing, that these Department of Space, Government of India's body will act like the other administrative work. They will take up the other administrative work. And for the privatization of space, this foreign direct investment has also been opened up. In this, up to 100% FDI will be there, and with certain limitations. Definitely, if you are making the complete set of satellite, then definitely it is capped to 74%. And if you are building some launch system in India from foreign market, then definitely 49% is the capping limit. Next is we have opened up one more, we can say, spaceport apart from the Sriharikota, which is in Andhra Pradesh. This is named as Kullasekhara It's in Tamil Nadu, because Sriharikota, in this particular segment, whenever we are launching any equatorial satellites, we have to cross the Sri Lankan part, because if the residual part of the launching vehicle falls in the Sri Lanka, it will definitely cause harm. So it has to be fall in the Bay of Bengal part. In order to do so, we have to maneuver this, and this maneuvering is known as dog leg maneuvering. But in the Kulasekara Pattinam part, we do not have to do this dog leg maneuvering, and the fuel invested in this maneuvering will be saved. At the same time, higher amount of payloads can be sent in the equatorial orbit with the Kulasekara Pattinam launch port. Next is the types of satellites. The launch vehicle that is in news is PSLV. PSLV, because it has 4 stages. First one is solid, then liquid, then solid, then liquid. These liquid stages contains Vikas engine. This Vikas engine is basically working very fine. PSLV in itself is known as the workhorse of the ISRO. But is in the 4th stage of PSLV, we have introduced some other engine known as PS4 engine. Recently, this PS4 engine, how it is different from the Vikas engine that is being earlier used. Vikas engine was very complex in nature. It was made by assembling too many parts. It will cause some of the wear and tear losses as well. So we have 3D printed this engine, and we have 3D printed this PS4 Vikas engine in the form of PS4 engine. So in that particular thing, PS4 is the 3D printed version. You can see it from here, that this is the actual Vikas engine with too many complex figures, and here you can see the PS4 engine with the 3D printed. The wear and tear losses will be minimum in this segment. So this is the part. The other is definitely your GSLV. GSLV is containing 3 stages: solid, liquid, and cryogenic part. So both of these liquid engines and cryogenic engines, they have achieved reignition as well. Means there is one more current information where we can say the achievement of reignition is very much important in order to reignite them while sending them back to earth, while falling in the surface of earth or ocean water. We can definitely reignite them, and we can means land them somewhere so that we can reuse them. So these particular segments can be reused. The other one is LVM3 or GSLV Mark 3. Both are the same name. It uses basically the same pattern of solid first stage, second is liquid, and the third is cryogenic. So again, this whole set of LVM3 for the Gaganyaan mission, it has been converted into HLVM3. This is human rated LVM3, where this portion has been added to it, which will, if the person the people astronauts are sitting in here, it will not face the direct frictional heat, and the temperature in this particular cabin will not increase while ascending in the space. The next one is SSLV. SSLV is launch on demand satellite, where the target of payload is 10 to 500 kg only to the low Earth orbit. So it is very good for SSLV to launch on demand whenever a demand comes to ISRO, and this is particularly being taken by the NSIL, New Space India Limited, under the Department of Space. We have just learned in 2019 we have made this. And the next part is RLVTD. This is relaunch vehicle, reusable launch vehicle technology demonstration, where this particular part is known as Pushpak. So this Pushpak will be reused each and every time. This is the relaunch vehicle technology demonstration, but later on there is one more satellite which is in the name of NGLV, new generation launch vehicle, which is now being termed as Surya. So NGLV Surya will be the future launch vehicle which have the capacity to uplift the payload 3 times more than the LVM3. So Kulasekara Patnam, we have already discussed about it. Vikas liquid engine, we have already discussed. SSLV is already been discussed. Reusable is discussed. So yes, This is one of the missions that was being in news in 2025 January and February. The SpaceX mission is Space Docking Experiment mission, where two of the satellites are launched in such a way that they can assign or assemble themselves in the space. Assembling themselves in a space moving at high speed all together is known as docking experiment. This docking is being done in the space while moving both of these two parts. One will be chasing and one will be the chasing the target. One, after chasing the target, it will definitely combine and couple up with the docking part. There is one more operation known as berthing. What is berthing? In many of the satellites, basically a robotic arm is being used whenever a particular target is there. Target is with having the robotic arms. Whenever this particular chaser will approach itself, the claws of the robotic arm will hold it, and by holding it, it can guide it to dock itself. This is known as berthing. So docking and berthing are the two specific operations. And again, there is one more thing that is in news, is the XPOSAT mission that is being launched in the 1st January 2024. What is this basically? XPOSAT mission. XPOSAT mission is launched by PSLV, and it is also being famous for the zero debris mission. What is this zero debris mission? Zero debris mission is that we are not leaving any part of unused part inside the space or above 100 kilometers of the Earth's surface. So it contains at the stage 4 a very special instrument named as POEM, PSLV or Vital Experimental Module POEM, which will give the accurate information of any left part being held in space, and it will help that particular part to deorbit and reach the atmosphere of the Earth, which will again, by the help of friction, it will be burned in the atmosphere. So in space, we are not leaving anything behind. That is why POEM 3 in this XPOSAT mission was very much in use. And POEM 3 in the XPOSAT mission, X-ray polarimeter satellite, is already in use, because this is one of its kind where in the world it is second in such number. Before that, NASA's IXPE is the only satellite which possesses the similar type of experiments. It will observe the distant stars, distant black holes which are emitting X-rays, and in that particular X-rays, polarimeter is also used, which will definitely narrow down the deflected information with that particular light. Now, the next thing is orbital debris mitigation. That is the same as we have talked about this XPOSAT mission. The next one is Gaganyaan program. Gaganyaan program basically includes this thing. What I may say, where is Gaganyaan? So Gaganyaan will come, no issues with that. But definitely Gaganyaan mission has been revised and added with one more form, in which we are also building our Bharatiya Antariksha Station. Bharatiya Antariksha Station is the international space station type of thing, which will be launched in the low Earth orbit. The first part will be done by December 2028. This is the target, and 4 missions of the Gaganyaan. Basically, the 4th mission will be the launching of Bharatiya Antariksha Station. It will be done with the NGLV part. So the next generation launch vehicle will also be developed till then in 2028, and definitely we will be launching BAS1. So apart from Bharatiya Antariksha Station, there is International Space Station already in operation, which was being developed by NASA, Russia, Europe, Japan and Canada. And in that, apart from this ISS, there is one Tiangong space station of China, which is much more developed than ISS, because ISS is a bit older. It is started building in 1998, while the Tiangong space station is basically launched in 2016 or 15 and operational by late 2022. And the other one in news with this space station is Gateway. Gateway is the process, or is an stage of Artemis program. Artemis program is basically dealt with NASA led with the help of Europe, Japan and Canada and other parts as well. So Gateway will be a space station in the lunar orbit. Always remember, it will not be in the Earth's orbit, rather it will be in the Moon's orbit or the lunar orbit. Then the other one is Axiom Station. It is also upcoming in nature. There is a company with the Axiom Space, named as Axiom Space in the USA, and it is basically building the commercial space station, which can be used by any of the players. So these are the versions. The next one is the Venus orbital mission. Venus orbital mission is again targeted for March 2008, which is commonly known as Shukrayaan mission. It will be the orbiter mission in nature. The key payloads are it is basically aimed at studying the atmosphere and the atmosphere and the surface with respect to structure and composition of the atmosphere and surface of the Venus planet. So Venus planet is already a sister planet in which 95% carbon dioxide in the atmosphere we see this, which is a huge amount of greenhouse gases, hence the temperature is too much inside it. And again there is like hydro or sulfuric acid clouds present over the Venus planet, and there are sulfuric acid rain as well happening in the Venus planet. And again the rotating period of this Venus is very slow. It takes approximately 224 Earth days to rotate once on its axis. And the future missions of other missions, we can say NASA's Da Vinci and Veritas, these are the two probable missions, and European Space Agency's Envision will be the other mission. This is the payload of Aditya L1, where various type of solar winds and photosphere, chromosphere, corona, these things will be definitely studied upon. This Aditya L1 placed in the Lagrange 1 point. Then the Chandrayaan mission, Chandrayaan 3 mission. The Chandrayaan 3 missions, as it achieved its success, we have got few of the output or the final output, as the lifts suggested that there is a presence of sulfur in the moon surface in the southern part of it. The CHASTE has suggested that the surface temperature near the south pole is approximately 70 degrees Celsius instead of 20 to 30 degrees Celsius, which we supposed earlier. Again, there is ILSA. ILSA had tested some of the moon quakes, not the earthquakes, but the moon quakes for the seismic activity and for the internal structure of the moon. And the other one is like APXS, SHAPE and Rambha LP. Definitely the studies of them will be further seen in here. There is also one of the rock known as Ferroan and Orthosite rock, fan rock in short, is being visible in the moon side, which definitely simplifies or suggests that the giant impact theory, that is the creation of moon after colliding it from the earth, there is a huge mass of like double the size of Mars or as same as the Mars size. It simply struck the earth at the very beginning, like 4.5 billion years ago. At that point of time, the complete mass is being subsumed inside the earth with the help of gravity, but a certain mass of it is flung distant apart with the formation of moon in the 4.5 billion years ago. So the Ferroan and Orthosite rock present in the earth of Canadian Shield and other very old Precambrian age portions, and the moon depicts that they both have been originated from the same thing. The Chandrayaan 3, yes. The next is Chandrayaan 4 mission. Chandrayaan 4 is again a very important mission, where it will be a sample return mission. And LUPEX mission is often considered as Chandrayaan 5. It will be in joint operation with ISRO and Japan. The next one is Chang'e mission, Chang'e 6 and Chang'e 5. These are the two missions of China on the moon side. The Chang'e 5 and Chang'e 6, both of these missions have been successfully completed in June 2024, where near side and the far side of the sample return mission has been successfully dealt with in Chang'e 5 and 6. So Proba 3 mission, there is one thing from the ESA, means European Space Agency, and the other is, no. There is one very famous mission is Proba 3 mission. Proba 3 mission is what it is doing is, suppose this is the sun, and sun outermost layer that we often see with our naked eye is known as photosphere. Around the photosphere, there is one more layer with the name of chromosphere, which is not at all visible to us. It is only visible when there is complete solar eclipse. When the complete solar eclipse happens, the outer ring, red glowing ring, will be termed as chromosphere. In order to study the chromosphere of the sun, the Proba mission is being launched. What it will do is, it will launch two sets of satellite. One will observe the chromosphere and the other one will act like as a simple, you can say, it will create a false eclipse for this satellite, so that it can study the chromosphere. So arrangement of these two satellites in sync is very much important, and it is being done with the help of NASA launch system. So although the Proba mission is from the European Space Agency, but still the launch system is being taken care by the ISRO. Next is Jupiter Icy Moon Explorer from the European Space Agencies and the Europa Clipper mission. These two of the missions are very much important to study the Jupiter moons. Europa Clipper mission is basically just subjected to the icy moon Europa, while the JUICE mission, Jupiter Icy Moon mission, will definitely cover 3 of the icy moons named as Callisto, Europa and Ganymede. Why it is so? Because these are the mysterious moons which have the maximum possibility of life inside them. Next is NISAR, which is very important for this year's prelims. NISAR is NASA ISRO Synthetic Aperture Radar, where we are building a synthetic aperture radar which can be termed as the most advanced radar system ever inside means this planet, for this planet. What it will do, it will be having a very strong band of this S-band SAR, synthetic aperture radar, sorry, L-band SAR. It will penetrate the vegetation surface, and it can look inside the earth and tracing the seismic activities as well. And it will be built by NASA, and ISRO is making S-band SAR. It is providing basically support to this L-band SAR, and all the launching part will be dealt with ISRO itself. So there is a joint mission, NASA and ISRO Synthetic Aperture Radar. The main objective is to study the environmental part and the seismic activities and the melting of the polar ice. Since this NISAR satellite will be launched in the polar orbit, it can definitely trace the melting part of snows in the polar regions. So this will be launched by GSLV Mark II, and this will be the best example of active remote sensing satellites. And apart from these active remote sensing satellites, then we in India we have RISAT and HISIS, hyperspectral imaging telescope or imaging satellite, or RISAT. RISAT is RISAT is imaging satellite, remote imaging satellite. RISAT is radar imaging satellite. So sorry for that. Radar imaging satellite. So after this, we come to our next part as Gaganyaan mission, and Gaganyaan mission definitely we will be having these 2 parts, and we can see this. The complete Gaganyaan mission will carry 3 astronauts named as Gaganyatris inside this crew module in the low Earth orbit. This crew module is being supported by this service module, where rest of the part of like oxygen providing, or the taking out of carbon dioxide part, or any survival part is dealt with service module, and rest of the microgravity experiments, all the equipments are placed inside or provided by the service module, and they all will be together placed in HLVM3 in here, and it will be launched to the low Earth orbit. It will revolve around the Earth in low Earth orbit for 3 to 4 days, and it will land safely in the Gujarat section of Arabian Sea. So this is how it will be done. These are the Gaganyatris. James Webb Space Telescope already talked about. Navik is again in news because of the 2nd generation satellites that is being introduced in the Navik system, that is navigation with Indian constellation. And here we have introduced a separate transponder which is of L1 band. Basically it is used for the wearable devices to track navigation, for using navigation in the wearable devices. Also this NVS-01 is very famous for using for the very first time the indigenously built atomic rubidium clock. Again, after that, information and communication technology is the part. Information and communication technology, the very first thing is artificial intelligence. We know that anything that can replace human brain is artificial intelligence, which do not involves the mindful or the thoughtfulness of the human brain is artificial intelligence. In very broader term, even your fully automatic washing machine is driven by artificial intelligence, because it can do repetitive work again and again and again without interfering the human mind setup, that when to switch it on, when to dry it, how many turns that it should take. So all these things where we are eliminating the human mind situation or the necessity of human mind, it definitely be dealing with artificial intelligence. there is one very crucial event which is known as machine learning. Machine learning uh, is an term or is a science or is a technology inside this artificial intelligence part, where if you provide this machine a set of data, it can definitely predict pattern on its own and definitely predict the outcomes of the future things as well. So this is how machine learning, but definitely it will deal in simple data as well, but only is the simple data. The next thing is artificial neural network, more precise than the machine learning. It basically imbibes or it basically mimics how brain thinks, human brain thinks. We often we often reach a decision by dividing the decision into multiple factors and assigning them particular importance numbers. Suppose you have to watch this lecture or not, it will depend upon the how much important this lecture is, who is teaching, which faculty is teaching, how much time is left for us. So all these things will be calculated, and then we will say that yes, we have to watch this lecture. So all these complex things are happening so frequently or so quickly inside our brain that we are not able to even think of these parameters explicitly, but this is happening inside our brain. So if the similar thing is being developed inside a software, it will be known as artificial neural network. And the next thing is, the next and the most or we can say the best part of artificial intelligence is deep learning. How is deep learning different from machine learning? Machine learning used to deal with the simple type of data, but deep learning will be training itself with the complex data like audio, pictures, voices, videos. All these things can act like a data. It do not require just numbers or text. It can even mimic the voices. It can create videos. It can create pictures. And definitely the best application of this deep learning we can see is generative AI. Generative AI, as we can see in here, that Modi ji is singing Gadar 2 song Main Nikla Gaddi Leke, and how this is possible, because Modi ji has never sung this song. It is just the mimicking of the voice. The next is video editing like Sora editing, then music making like the Suno AI, and there is plenty of playlists you will find on YouTube with Muhammad Rafi AI covers, where the voice of Muhammad Rafi has been cloned for the recent songs. Similarly, there is a voice cloning that was plenty in news. Along with that, the Nobel Prize goes to John J. Hopfield and Jeffrey Hinton. That is why, for their work in the discoveries and inventions that enables machine learning with artificial neural network, for which we have studied right now. So again, there is one more thing like spiking neural network SSN, or there is one more thing that we often see it or call it as neuromorphic computing. As artificial neural network uses software, it uses basically chip or hardware. So this is the basic difference between these two. About deepfakes, we have already heard about the newses of this what the name of the actress, Rashmika Mandana and Katrina Kaif, being imposed by some other bodies, means their faces were imposed on some others' bodies using some obscene activities which was not for the public to see. So that are the deepfakes. Deepfakes basically clone the faces and expression of a particular person and clone it on any of the faces or any of the body. So swapping faces can be termed as deepfakes, and definitely it is a punishable offense under section 66D of IT Act 2000 and under the rules of 31B and 32B of the IT Ethics Code Rules 2021. Then there is a meaning in GPAI having India as the lead chair. India is having the lead chair in GPAI this year, and New Delhi Declaration, if the UPSC asks that from where it has been related, you can definitely tell that it is related with AI. And again, with the Bletchley Declaration and Hiroshima AI process, the same thing, they are discussing about the artificial intelligence. DeepSeek part is again very much important, because there are few scientists in the China which does not require the hard processed graphics processing unit or the very smart graphics processing unit, high end graphic processing unit, in order to train their artificial intelligence, or in order to train their LLMs, that is large language models, just like ChatGPT, Gemini, Grok, and so on. So DeepSeek is just that revolution which brought Nvidia shares down in a single day when they think that this GPUs are not at all required. The next one is quantum technology. In quantum technology, the very basic thing is we are using qubits instead of binary digits. Binary 0 and 1, we are using qubits. We have to learn about the quantum mechanics. For this quantum mechanics, we have to learn about the key principles. Key principle is, the first one is the quantum superposition. Quantum superposition says that a qubit can be at both 0 and 1 place at the same time. It can be 0 and 1 at the same time. And again, there is a quantum entanglement. The quantum qubits definitely occurs each and every time in pairs. And once entangled and separated apart in any of the distance, they will always be connected with each other. Then quantum interference is in the similar way as the light happens for the interference. Quantum tunneling is that ability of the particles to seek through a particular wall or a particular barrier, which we call it that it will never pass it, but it will make a tunnel and then it will be passed. We will find the particles in the other side as well. So this is the special part of quantum tunneling. Basically, these things are used in quantum computation, quantum communication, and magnetometry and atomic clocks, quantum materials, thematic hubs. These things are being created. We are definitely launching various quantum chips so that we can have these chips inside our computer system as well. Recently, Microsoft Majorana 1 was in news with this chip, and there is again some Amazon Web Service Ocelot, Google's Willow, and IBM Heron was in news. Next is supercomputer. Supercomputer was not that much in news. We just have to know that there are two new computers named Arka and Arunika, which has joined the league of top 5 computers from the Indian part that will be used in the meteorological researches. And the next one is mixed reality technology. Just in the say that augmented reality is something that is viewed upon or added to the screen. Suppose you are using the camera in the beauty version. You will see rouge and lipsticks or kajal or hairband or cap. All these things are happening inside the mobile phone. Means in the real world, you are adding something to it that is being augmenting something. This is known as augmented reality. This is not an immersive experience. You will not just lost inside the virtual world, but it will happen in the virtual reality where you will be having a VR headset, and in this VR headset, you will be completely immersive in that particular environment, and a virtual world will open up inside you. So this is the virtual reality. And when we merge both of these things, we come to mixed reality. Just suppose a Vision Apple's Vision Pro. So this is the best example for mixed reality where we will be having both of these things. Other thing is cloud computing, and cloud computing comes with a service. Now, the infrastructure or software or platform as a service is being old. Nowadays, there are graphics GPUs as a service. Suppose you have bought a very cheap mobile of 6,000 or 7,000 with very or no GPUs inside it. So the processing will be very problematic. What the GPUs do? GPUs are basically graphics processing unit. Apart from CPU, which are central processing unit. Central processing units basically worked on the sequential method. Sequential method means that if it is allocated with 100 of tasks, it will perform each and every task one by one in 100 times. Suppose for the GPU, if you allocate 100 tasks to it, it will definitely divide these 100 tasks into 10 different tasks of 10 bunches, and executing all the 10 bunches all together, it means that parallel processing is possible in the GPU. That is why the processors can handle lots and lots of data. And that is why it is being used in the artificial intelligence training, LLMs trainings. And at the same time, for the 4K videos playing or the very heavy games playing, for the actual graphics to work, this graphics processing unit is used. So the next thing is blockchain. Blockchain is also being used as a service nowadays, and BaaS as a service. And Microsoft Azure's BaaS is the example. Even India has also launched it with the name of Vishwasya blockchain technology stack. So next is nuclear technology. Nuclear technology, the very basic thing that we have to know is, for the very first time we have approached this fast breeder reactor. Means the famous, our beloved Dr. Homi Jahangir Baba Sahab has already told the three-stage nuclear power program. In this three-stage nuclear power program, what we did is producing the electricity with the help of thorium, which is already being already present in India in too much of quantity. We say that 25% of the complete thorium reserve resides in India, and that is why thorium, being the fertile material, not fissile material, the fertile material should be converted into fissile and then it will be used. So it cannot be used directly. Hence a three-stage work. We have started in the stage 1 part from the very beginning, and from that part we just moved to the second stage in 2024, where the first form of fast breeder reactors are there. Why the name fast breeder reactor? Fast because we definitely can use fast moving neutrons and there is no use of moderators, which slows down the neutrons. So no need of moderator inside it. And the breeder reactor is because the plutonium-239, which is the byproduct in the stage 1, is being used along with uranium-238, and again in the byproduct you are getting plutonium-239 again. So plutonium-39 means more it breeds than the fuel it consumes. That means it is a breeder. That is why the name breeder reactor. And in the third stage, the final product there was uranium-233, and this uranium-233 again uses thorium-232, and this goes in cycle. That is why the thorium is directly used in the stage 3, and it will happen somewhere in 2035 or 40. China has already been done this thorium build. Means it might be possible that in this year in China can use thorium based reactor. Then the next one is not this. Then the next one is fast breeder reactor. We have already talked about it. Fast breeder reactor. Then is something known as small modular reactors. What are those small modular reactors? Small modular reactors basically work differently from the large conventional type of reactors in a way that they are modular in nature. Means they are manufactured in the industry or in the manufacturing zone, then transported to the place where it has to be installed, and they can be assembled just like our kitchen, modular kitchen, where each and every steel baskets are being manufactured and made in the factory itself, just arrives at our home, and the carpenter used to fix them or assemble them in our kitchen. That is why the name modular kitchen. The similar way will goes in here as well. Is small because the power of the electricity production will be capped at 300 megawatt. Means up to 300 megawatt electricity capacity will be there. Creating these things, the India has launched its first Bharatiya. Here you can see this. A small modular reactor with the name of Bharat Small Reactor. Where it is? Here, Bharat Small Reactors. So it will be like pressurized heavy water reactors with 220 megawatt power productions. So there is a target as well. We have our Bharat Small Reactors with a basic target. What is this target? Target is in here. These targets symbolizes that we have to produce at least 5 of them. Here you can see this Bharatiya Energy Mission. No, it has not been mentioned in here, but definitely there is a mission. This is known as the Nuclear Energy Mission, and under this 20,000 crore rupees has been given in this budget for us, that till 2031 we have to build 5 small modular reactors indigenously. So this is the target given. Next is the tokamak reactor. Tokamak reactors, rest of the reactors that we are discussing till now is fission based reactor, but this tokamak will be fusion based reactor. The ITER along with so many countries with India, not in India, it is basically produced in France basically. So European Union is investing a lot in it. But apart from European Union, US, Russia, Japan, China, Republic of Korea, these countries are also investing a lot in this ITER with the tokamak reactor. And in this, the objective is to have high fusion power gain which is greater than 10. Means whatever energy we are giving it as an input, the output will be at least of 10 times. Suppose if the 500 megawatt energy is being produced, then minimum we have to spend like 50 megawatt of energy. That is why achieving the high fusion power gain of more than 10. But still now what we have achieved is 0.67 only. Means we are giving more energy and receiving less. So tokamak is again started in India from 1985s as well. So 1980s we are making tokamak Aditya. The whole ITER India project is basically dealt with Institute of Plasma Research, and again the Aditya is already being developed by the IPR. The next is world's first CO2 methanol plant, and CO2 methanol plant, what we can say in it. Yes, this is simply a conversion of carbon dioxide from the flue gas to methanol. Methanol is the simplest alcohol form, methyl alcohol, wood alcohol. It will be used in the blending of fuels, and it will be used as a lubricant and as a diluter, I can say. So these things can be used. Next is alternate energy, where we can see sodium batteries, which will be more capable of charging. Means it charges very fast, and it will operate on higher temperature, and it will be safer to use. It will be charged in seconds rather than lithium ion batteries, which takes, I think, hours to charge. The last chapter is space technology. Defense technology, where we will see the Agni 5 mission. Agni 5 mission has been launched with mission Divyastra, which conducts with MIRV technology. This MIRV technology is multiple independently targeted re-entry vehicle, where we can target single-handedly A, B, C, D, E various positions, and we can load multiple warheads, and with firing of a single ballistic missile, we can target multiple targets. Earlier, multiple warhead carrying capacity was there, but the target part was not there. It will be distributed. Multiple warheads will be striking the ground in multiple places, but not planned in and around the place that we target. But now with MIRV technology, we can definitely target these things. By doing this, India becomes the 6th nation after P5 to have indigenously built MIRV technology. The other one is BrahMos NG, the BrahMos 1 and 2. After BrahMos 1 and 2, there is BrahMos NG, which will be like 290 kilometers of range. Along with, it is 50% lighter in nature, so that it can be used in the aircrafts as well. It is 3 meters shorter in nature, so that again it can be used for the aircrafts. Even we are using it for Sukhoi 30 MKI, and it will be again used for export version as well. The speed will be like 3.5 Mach, and hence it will be of 2 stage. One is solid stage and the other one is of ramjet. Again, from BrahMos 2, if you are talking, BrahMos 2 goes to the speed of 7 to 8 Mach. So 7 to 8 Mach, it will use scramjet engine instead of ramjet engine. You have to remember this. Then again, there is air-to-air missile, Astra missile is there, and we have renamed the Astra Mk3 missile as Gandiv missile. Again, there is ballistic missile defense for the air defense part. We are using the dual layered ballistic missile defense part. Prithvi air defense and advanced air defense, which will counter the ballistic missiles coming from the enemy side in our country. Next is Pralay missile and Pinaka rocket. Pralay missile is quasi-ballistic in nature, where it can go to a limit and then it can change its path for once in mid-air. This is Pralay missile, quasi-ballistic missile. And one is in news is definitely the S-400 missile. S-400 missile is imported from the Russia, and S-400 missile is canister based and portable device, where in one single canister we can launch up to 4 missiles, which can target in different directions. Means altogether in 3 canisters we can acquire 12 targets at a single point of time. So without wasting any time further, it can target 12 targets simultaneously. Maximum speed is 40 Mach, which is a lot more than 29,000, I think 29,000, 30,000 kilometer per minute. Just calculate how much it is. It is basically 14 into 1, 2, 3, 4 kilometer per hour. You will get the information that how much it is. So normal range is 250 to 440 kilometers. The 3 has already been adopted. The next one is submarine in news. There are few submarines which are inducted recently in the SSBN category. That is, it has been inducted with nuclear power, along with it has the nuclear warhead capability as well. So INS Arighat and Aridaman are the two of them, having K4 and K15 Sagarika missiles. I'll talk to them. I will talk for them later in just a while. The other is SSK class submarine, which is diesel power submarines, and recently INS Vagir and Vakshir are from the Kalvari class submarine that has been inducted recently inside our navy. So in the submarine part, you can see the K4 missile, nuclear capable ballistic missile, has range of approximately 3,500 km, while the K15 missile, the Sagarika missile, has the capacity of 700 to 1,900 kilometers. So they have the capacity to carry this nuclear warhead along with them. And with this, we came to the end of this session. I hope this is very fruitful for your revision, and the quick revision part was of too much use for you. This is it from my end, and definitely, as I have mentioned in the earlier part as well, that you can reach and you can get this PPT. You can reach this PPT on the Telegram channel in here. I have already told that you can search the Telegram channel with the name Shivam Sir PW in here. Yes, you can get this PDF here whenever it is uploaded on the YouTube channel. So that's all from my end. I hope in best of your memory, it will remain till the exam, and you can utilize this information inside your examination hall as well, and definitely it helps you to score more in this prelims. That's all from my end. Thank you very much for listening it very patiently, friends. Have a nice day and all the best for your prelims exam. Thank you very much.